Hello everyone, we are group 4 in freshwater biology course. The instructor is Dr. Michael Bari. We are going to cover about the field to Al-Jabal Al-Akhdar reservoir, which are small man-made water bodies constructed to provide a non-potable water source for nearby villages. Although simple in structure, they represent important freshwater habitat in mountainous and arid regions. Studying these reservoirs is essential because they allow us to understand how physical, chemical, and biological factors interact in a standing water ecosystem and how these systems support biodiversity under limited water conditions. The aim of this practical was to investigate the ecological characteristics of one of these reservoirs by measuring key water parameters, assessing water clarity and nutrient levels, and collecting biological samples from different zones. The site is located at high altitude in Jabal Al-Akhdar. It was approximately 2,000 meters high, where color temperatures, as I saw it from the phone, it was 19 Celsius. Terraced agriculture in the limited watershed inflow influence water quality and ecological processes. This unique environment makes the reservoir an ideal natural laboratory for studying freshwater ecology in Oman. Moving now to the sampling methodology. In the beginning, groups were divided into two groups. Each group has their tools to use. We described the site and mapped it. Later, we used the Secchi disc. Secchi disc was lowered vertically into the water until it disappeared from the view. Secchi disc is for water transparency measurement. The depth of disappearance was recorded as the Secchi depth, the ZM. The measurement was repeated 3 times and at different points along the reservoir wall to obtain an average value for and also accurate for the turbidity of the reservoir. The vertical sampling of the zooplankton, Schlender batalostra. We used this with as it's shown in the photo. It is 4-liter volume was lowered at half to 1 meter intervals from the surface to the bottom. At each depth, the trap was triggered to collect an isolated water sample. Samples were transferred to labeled jars and preserved with 70% ethanol for later zooplankton identification and counter. We also used the vertical water sampling, the Van Dorn sampler. Water samples were collected at half-meter intervals using a Van Dorn sampler. Approximately 400 milliliter from each depth was immediately used to measure temperature, dissolved oxygen, and the pH to avoid changes caused by the exposure to the air. Also, an additional 2-liter surface sampling was collected and labeled for alkalinity analysis. Reactive phosphorus was measured on site at 1 meter interval using a chlororimetric test kit.
Now we continue to our results, and also the parts of the methods. That's what we did in the laboratory session. What we did in the laboratory, especially for our group in week 2, group 2, we take our sample, which is the center of test samples with a different depth. We take it to the laboratory and we measure it under the telescopes to see how much the species in each depth and what's the type of species that we have in this types of lake in Jabal Al-Akhdar. And also the other part, which is the chlorophyll absorbance. We use a spectrophotometer to put our samples there, the different depth, also to know the absorbance of the chlorophyll related to the depth of the lake. And now we will go to our results. The result shows a variety of different species, but most of them was cyclopoid and cyclodaphnia, and also a little bit of a small daphnias, which we measured. Also, there was a difference in the depth. It was increasing by the more depth. When we go to the more depth, we see the more zooplanktons occur in our samples by counted in the telescopes. So we see that there's the replicate 1 with a variety of different cyclopoids. Going to the replicate 2, we see it's also increasing with the depth, and especially as we say cyclopoids and cyclodaphnia. And also the pH is also changing with the depth, which is a little bit decreasing in the pH when we go further to the more depth. Also the oxygen, as we know the oxygen, and as we measure it, the oxygen is increasing with the depth, so there is a less oxygen in the depth. And we will not go much details because it's a results part. And also in the chlorophyll, we see the chlorophyll its absorption is changing with the depth also. The absorption with the depth is increasing a little bit, especially for chlorophyll A profile, which we did in the spot spectrometer to measure the absorption.
Okay, now to go to discussion. First, say the zooplankton distribution with depth. We start with the cyclopoid, where the dominant group at almost all depth, with especially high number at deeper layer from 4 to 5 meters. This indicates their ability to tolerate low light and potential variation in oxygen or temperature. Another thing, the Seriodiphenia were present but in much lower abundance, suggesting they are less competitive under the reservoirs condition. About the Daphnia, were extremely rare or absent across most depth. This may indicate predation, pressure, or unstable physical condition in surface layer. Now we move to the other points. The relationship between zooplankton and light availability. The measured Secchi depth from 1.35 to 1.5 shows that light penetration is limited. Science cyclopoid were still abundant below the photic zone, larger than 2 meters. It suggests they are not strongly dependent on high light condition. Reduced light may limit phytoplankton growth, indirectly affecting herbivorous zooplankton such as Daphnia. The second thing, or the third thing, the chlorophyll A concentration. Chlorophyll A values were very low overall, approximately from 0.1 to 0.3 microgram per liter. This value indicates low primary productivity and classify the reservoir as all bigtrophic based on algae biomes. Lack of clear peak in chlorophyll A at any depth suggested no strong vertical stratification of phytoplankton. Okay, the fourth thing is stratification the reservoir. Based on zooplankton distribution and the relative uniform chlorophyll A values, the reservoir appear weakly stratified or not stratified. Light limitation and shallow depth likely prevent strong using the Secchi depth. According to standard Secchi-based classification, trophic state Secchi depth, we have 4 depths. The first, oligotrophic, that is larger than 4 meters. The second is mesotrophic, which is between 2 to 4 meters, and the eutrophic, which is from 2 to 1 meter, and the hypertrophic, hyper-eutrophic, which is less than 1 meter. Okay. The reservoir Secchi depth from 1.35 to 1.5 suggests eutrophic condition. However, chlorophyll A level are very low in oligotrophic. This mismatch indicates that the turbidity is likely dominated by non-algae particles such as clay, silt, or runoff from surrounded rocky slope rather than algae bloom. Okay. The second thing is difference between secondary platel sampler and plankton nets. The secondary platel sampler gives precise quantitative measurement because it samples an exact known volume 4 liter. Plankton nets collect much larger but less defined water volume, so density may appear high and less accurate. But what the advantage? Such secondary plates accurate density, depth specific sampling. A plankton net better for collecting diversity, especially rare taxa. Okay, now we go to the conclusion. The conclusion say the reservoir does not appear strongly stratified, as both chlorophyll and zooplankton showed relative uniform vertical distribution. Overall, the system reflects a low productivity mountain reservoir influenced mainly by physical factors such as limited light penetration and suspending sediments. Thank you for listening, and if you have any question, let us know. Thanks.